Hello, everyone. Welcome to Career Pathways in Microbiology. My name is Shilpa Gedwa, Program Officer in the Education Department at ASM. We're very excited to have you join us today. The purpose of today's webinar, Career Pathways in Microbiology, is to bring you awareness of the different types of careers that exist in microbiology in hopes that you find something that interests you. Today, we have three speakers with us who work in climate change, and we have our moderator, Benjamin Liu.
Hello, everyone. Welcome to the ASM webinar on climate change, the career paths in the climate change. My name is Benjamin Liu. I'm today's session's moderator. I'm an assistant professor from George Washington University and Children's National Hospital in Washington, D.C. So today, it is our privilege to have three speakers. Each speaker will give a 12 minutes presentation. Now I'm gonna introduce the first speaker. Her name is Arlene Borby. She is a research agronomist at the United States Department of Agriculture Research Service. Her research focus includes the water resource management, sustainable intensification of rice cropping systems. Arlene, I'll turn the floor to you.
Thank you, Dr. Liu, and good afternoon, everyone. So my name is Arlene Adviento Borby. I'm a research agronomist for the Delta Water Management Research Unit. It is under the United States Department of Agriculture, Agricultural Research Service Unit of the federal government. There.
Okay.
So
Yeah,
in addition to the research agronomist position, I also hold two positions globally. I am a lead scientist for the United States representative under the Global Research Alliance on Agricultural Greenhouse Gas Emissions for the Paddy Rice Research Group, American Group. I also hold as a counselor for the Conservation Agriculture Alliance for Asia Pacific, and this is for the Food and Agriculture Organization, United Nations. With this study or with this type of positions, I basically work on many aspects of rice research and agricultural production projects. But before I go on to that work that I've been doing, I'd like to introduce to you that I am not a full-blooded United States citizen. I came from the Philippines. And basically in the Luzon, the biggest islands in the Philippines, in the Laguna province. And the Philippines is in the Southeast Asian region, and Laguna is right at the heart of that biggest island in the Philippines. If you don't know the Philippine island, it is very famous for its rice terraces in the Banawe province, the underground river in Palawan, and of course the Boracay Island in the Clan. With regards to my career pathways, I started my bachelor's degree in agriculture in the University of the Philippines in Los Baños, Laguna. My major mainly is in agriculture. After my bachelor's degree, I graduated a soil science major. I worked on the International Rice Research Institute, and that is also located in the Philippines, as a researcher initially, and then climb up to assistant scientist under the supervision of soil scientists in rice at Erie. And while doing my work at Erie, I also took advantage of a free tuition fee for master program. So I took Master of Science in Soil Chemistry with the Erie special program and the UP Los Baños master program. After completing my master's degree, I went to the University of Nebraska Lincoln because they offered the scholarship for PhD program, and I took a PhD in agronomy. After finishing my PhD, I worked as a postdoc in Penn State with the Department of Agriculture and Biological Engineering. In this work, I use mainly my skills and experience in rice and crop research and greenhouse gas emissions. I was able to get that job. And finally, I want to go back to fully rice research. I took a job as a project scientist at the University of California in Davis, specifically in Department of Plant Sciences, where I worked with the Mars project on greenhouse gas emissions, rice, and fertility in California rice. Looking for a much more permanent position, I applied as a research agronomist for the USDA ARS in Jonesboro. And in this position, I started working on much more broader aspect of crop production, water resources projects, and also looking into the possibility of developing a climate smart cropping system in the lower Mississippi River Delta. Drivers for my chosen career. Basically, these are fourfolds. One is because of free tuition. There are a lot of very productive programs on agricultural research. So I took advantage of that free tuitions and also the possibility of engaging towards much more international collaboration because I worked with many scientists in the past, specifically those that have real impact in terms of rice research and soil research, such as Dr. Akim Doberman, who used to be the director of Rothamsted Research in the UK, and now he's the lead scientist of the IFA, or International Fertilizer Association. Guy Joseph Kirk also is a great influence to me towards soil research, and of course, Kenneth Kasman, who is a food prize winner in terms of yield gap analysis in University of Nebraska. Finally, it's because of my real passion in terms of moving forward into the 21st century in agricultural research, because food is definitely very important in our world, and also the degradation of environment is also accompanied by food production. So I'd like to focus on much more in working towards that direction. And finally, because all of my tuitions were free, from bachelor's to PhD, I'd like to return the favor. What do I do? Basically, I do study crop and water management practices in the lower Mississippi River because that's my assignment. In addition to that, I also work on technologies like building and develop methodologies, instrumentations, analysis, data set gathering, and looking into much more impactful outcomes of all agricultural research. So I don't just develop or make ideas, but also make this project usable to farmers, public, and specifically to all other users of the cropping systems that I am developing. So these are some examples of the work that I've been doing in Arkansas and also in other countries. Mainly, I worked on what the farmers have been doing in their fields and then tried to optimize that. Whatever is available in their field, I don't develop new ideas that farmers cannot use. So they are just basically a practical technologies that we are refining. I also have a project with Japan right now, which is looking into the much more detailed understanding of greenhouse gas emissions from two different varieties that are very popular in the States and also in Japan, and looking into the danger of food nutrition, such as heavy metals in the grains, because these are very common if you change the management of rice. In addition to that, we also would like to protect water resources because right now we have issues on water availability. Aside from rice, I also have another project, which is corn and cotton. And this time you're looking at cotton field, which is a long-term application of conventional practices, saving the environment while sustaining the cotton yield with a Cotton Incorporated. This is funded by a public grant office, and they are looking into refining whatever are conservation practices available in the region and try to sustain its lint yield at the same time lowering its carbon footprint. I was also summoned to work into a big collaborative project wherein we are looking at the impact of climate crisis, such as high night temperature, on the availability of high grain quality and also grain yield in rice, because once we have this fluctuating high night temperature, it will impact the grain yield, and that is the one we are trying to look at, whether there are some traits that can we use and include it to a new plant type to be able to have a high tolerance in high night temperature. With this project, we discovered 4 genes that are influencing the grain yield of rice. Okay, and also I do a lot of instrumentations to measure greenhouse gas emissions and validating it. and making it much more meaningful for IPCC reports. I do have collaborations locally, farmers, professors, students, nationally with private companies, big corporations such as Kellogg's, Mars, Google, Grantham Foundation, and internationally because of those positions I am holding right now. More especially those countries that the major staple food is rice, and of course federal agencies. As a federal scientist, I have to work with federal agents to be able to come up with meaningful objectives and goals of our projects towards increase in food production and following on preserving environmental degradation. Currently, I do have staff and students working for my project. They are coming from all over the world. I have one from Tanzania, from Japan, from Nepal, from the Philippines, and we are all working towards one common goal, which is the passion in understanding the impact of this climate crisis on rice and cereal production. I also have collaboration with national and international universities and coming up with what much more understanding of the project that we are doing, also sharing information on what they have found out in that country or what they have gained from that kind of technology, and try to swap our information from one region to the other. In terms of a day in the life of an agronomist, it's very complicated, especially during the summertime, because that's when we have our peak of our activities. I don't work in the office only. I work in the field, and I also serve as a resource person to whoever wanted to understand greenhouse gas emissions and rice production. So in the morning, I often go to the field, look at my sites, determine if there are issues involved, especially in field operations. If I have guests coming from other countries or other companies, I do show them our field experiments and have them understand what we are doing in a practical way. I do also have students after going to the field. I work with my students, looking at their data, problems in methodologies, or issues in terms of financial research work. And as a SY scientist, I also have to deal with administrative work because I do have some students and staff with me. So I have to follow up on administrative matters. And if there are issues that we need to comply with the HR, then I also do that. And then in the afternoon, I do have another meeting with my colleagues and collaborators in the States or sometimes in Latin America and discuss about our future meeting or workshops that we plan to do for the next year. In the afternoon, if I still have some time, I do write the research projects and papers. And sometimes I go to the lab and check if our instruments are working or some issues in the supplies that we ordered, or scientific items that we need for certain laboratory or field measurements. And finally, even at night, I do work as well because my collaborators are not only in the States, but also sometimes in Europe or sometimes in New Zealand, and time difference is very difficult for those people. That's why most of the time I'm the one sacrificing my time and stay at night because all of them are in the other side of the world. So that's a typical day of my work. And with that, if you have questions, let me know. Thank you so much for your attention.
Thank you, Arlene, for your nice presentation. Our next speaker is Charles Brook. Mr. Brook is the program lead of Spark Climate Solutions. He leads Spark's livestock enteric methane mitigation program, building off his science, policy, and the practice expertise in the field. He also served as the coordinator for the Globe Ethane Hub Enteric Fermentation Accelerator Science Oversight Committee. Mr. Brook, I'll turn the floor to you.
All right. Wonderful. Thank you very much. So as mentioned, Charles Brook, program lead for enteric methane at Spark Climate Solutions. Really, I'm just a microbiologist at heart who's found a way to use microbiology in a bunch of different areas, and ended up landing directly in enteric methane, or cow burps, and how methanogens produce methane in the rumen of cows. But it wasn't always that way. I will say, originally I was not geared for path in academia. I started life out as a stonemason. Started laying stone, brick, and block, as well as general construction and labor. And it wasn't until I started actually brewing beer at home, read John Palmer's How to Brew, and learned about yeast and how they function, and tried to understand more about the enzymatic pathways of fermentation, I really got interested in microbiology. And so from there, I had a major shift, went to college up in Northern California at Chico State, home to one of the best breweries in the world, maybe if not the best brewery in the world, Sierra Nevada. And started really working on microbiology and getting involved in undergraduate research. Really critical aspect of everything I've done has started in the undergraduate research phase, and started really in environmental microbiology, even though my original track was clinical microbiology. Started off in clinical laboratory science, and then got pulled into the dark side of environmental research because it was much more open field, a lot more unanswered questions. Started looking at iron oxidizing bacteria and reducing bacteria. This is a picture of this red iron rust in these waterways here on the right. And then looking at how we can apply microbiology in the real world, beyond environmental impacts. Worked in food safety, built the food safety plan when FSMA was passed for our university's meats laboratory, and learned about processing and handling meat and livestock. And during that time, also learned bioinformatics and metagenomics. Is when back then when 16S sequencing was all the rage, and applied that to different types of anaerobic digestion. And this is a picture of the first food waste digester built in California. We applied metagenomics to understand microbial succession and feedstocks optimization in there. And so I had experience in livestock, I had experience in anaerobic digestion and methane and metagenomics. And it was at my local ASM, Northern California American Society for Microbiology meeting, that I met an individual who was working on biomass conversion, and was actually trying to find enzymes from the rumen of cows, which break down biomass and convert that into energy, and trying to use those enzymes for more efficient biofuel production. And that's when I moved into looking at the rumen microbiome. It's an amazing complex system of bacteria, protozoa, fungi, viruses, and it produces a significant amount of the world's methane. Most of it is eructated, burped out of the animal's mouth, and not the other end, as is often misconstrued. But it is the rumen microbiome really is the key aspect driving all methane on planet Earth. Well, excuse me, the rumen microbiome is key for livestock. Methanogenesis is the key to driving all methane on planet Earth, including the fossil methane that was originally produced by methanogens a long time ago. But really understanding the rumen microbiome took me on a real whirlwind. And everything from trying to understand the enzymes themselves to lab bench, to taking rumen fluid out of animals, testing different compounds, how they worked, and then moving things into animals. And that's when we started working on enteric methane mitigating inhibitors. And so how can we inhibit methanogenesis in the rumen? And I've been studying these compounds for quite some time now. So for context, today we're on a track to try to limit warming to about 1.5 degrees C. That's the goal, right? We want to try to limit warming. We know we're warming. We want to try to limit it to under 1.5 degrees C of change. And right now, only livestock enteric methane makes up about 0.1 degrees C of the change that we feel. By the end of the century, it's going to make up about 1/4 of a degree Celsius. And that's about 460 gigatons of CO2 equivalent. Right now, the technologies that we have available for us to mitigate that methane only really covers about 10% of those emissions, and we need to be able to find more. So part of my career was doing really early work on some solutions in this space. So feeding seaweed to cows because it has a methanogenesis inhibitor called bromoform included in it. And that took a long progression into a lot of different areas, all the way to what we see now. So there's a picture on the bottom right. Can CRISPR cut methane emissions from cow guts? And that was my old mentor, Matthias Hess, on the left, holding a stainless steel vessel that I built in my garage to test for can we use CRISPR to reduce methane production from livestock. But this took quite some time, and being on the edge of a lot of this research wasn't very easy. And it took a while for the world to really start to understand the impact of livestock enteric methane and what we needed for solutions in the space. And so I worked in industry. I will say that funding wasn't when you're on the edge of research, there's either two paths. Either you have great funding availability because you're on the edge of research, or no one understands what you're doing and funding is not very available. And unfortunately, we were the latter. And so it took a couple of years for things to develop. I joined California, who was a very forward-looking state in livestock methane, as the lead scientist for their short-lived climate pollutants program. My work on digesters applications, work on manure management, and then building the enteric methane research programs for the state of California. And these were wonderful programs, all focused on really understanding how methanogenesis operates within the rumen. And we started to see a lot more traction build up. We held, and I still organize the only annual conference on enteric methane emissions globally. And now there's global efforts to try to increase the research on for research and development, including the Global Methane Hub's Enteric Fermentation Research and Development Accelerator, in which I serve as the coordinator for the Science Oversight Committee. And that brings it to why I joined Spark, and a little bit about what I actually do in this space. Is Spark is a nonprofit organization. We are a field developer. We try to understand underserved climate fields. So if we were to look at where solar was 60 years ago, or carbon dioxide removal, or any other climate mitigation field, and try to understand how could we have developed these fields faster, or how can we develop these fields faster? And that's what the approach we take with enteric methane. Where are we at today? What tools do we need? And how can we approach the field and develop it faster so we have the tools that are necessary to mitigate its climate impacts? And that includes working with scientists, working with policymakers, working with businesses and startups, nonprofits across the board towards realizing the potential of these strategies. So why am I doing any of this? Really, I really enjoy the complexity of these systems, and there's no more system more complex than I will say microbial communities of our food systems. Those are probably two of the most complex systems in our world, and trying to put them together is very challenging. Our work is at the intersection of R&D, policy, communications, and business, and microbiology is inherently in the center of all these technologies. More or less, I don't enjoy sleeping, and I don't do much of it because I love what I do. And some things I had a lot of angst and consternation when I was early in research, trying to understand the role of these different technologies and why no one would support them. And I can't. I will say that I can't complain that things aren't happening as fast as I would like them to now, because I'm responsible for trying to help make them happen. And that's a lot of responsibility, but also a lot of freedom. And so love what I do, trying to push this ecosystem forward, communicating the science to whoever needs to listen across the different sectors. And I would show you what I do every day, but my calendar is an absolute mess. So I don't know if it's really a lot of engagement. I talk and I meet with a lot of people. We write a lot. I travel quite a bit around the world talking to people because this is a global issue. And working with even directly on farms, spend a lot of time on farms on different technologies, in the lab, trying to support different technologies across the board. So with that, I think we're going to save Q&A for afterwards, but I can jump off of this here. Thank you.
Thank you, Charles, for the nice presentation. Enjoy it very much. Last but not least, we have the third speaker, Mamta Rawat. Dr. Rawat is the program director in the division of organismal systems in the bio directory of NSF. She also served as the science advisor in the bio directory of NSF. Previously, she was a professor in the California State University studying stress responses of bacteria. Dr. Rawat, please.
Thank you so much. First, Dr. Du for that introduction, and ASM and Shilpa for organizing this session. And good afternoon, everybody. Let me tell you a little bit about what I do. And before I start with that, I will tell you a little bit about me. I was born in the foothills of the Himalayas, right here. I don't think I have a pointer, so you can just have to see. And from there, when I was 9, my family moved to Montreal, Canada. So I grew up in Canada, and I did a Bachelor's of Science in Biochemistry at University of Toronto. Then at the University of Toronto, I actually did undergraduate research, as Charles mentioned. Undergraduate research is one of those experiences that is sometimes transformative, and you'll see how it transformed my life. So I performed undergraduate research on sea lampreys, which are a major nuisance in the Great Lakes. After that, I got a PhD in plant physiology and biochemistry from Louisiana State University. And one of the reasons I switched from what was really a cardiovascular lab at University of Toronto to plant physiology and biochemistry was I did not want to work with mice. So my degree, my PhD dissertation is on the carbon dioxide concentrating mechanism of algae. I worked on Chlamydomonas, and this has implications for photosynthesis and how to help with increasing photosynthate. I also was a postdoctoral scholar at University of British Columbia in the Division of Infectious Diseases in the School of Medicine, and also at University of California, San Diego in the Department of Chemistry and Biochemistry. And here I again switched organisms. I worked on bacteria. I worked on mycobacteria. You may have heard of Mycobacterium tuberculosis, which causes tuberculosis. That is a disease that affects many people in the world. In fact, 1/3 of the world is infected with this bacterium. And about 10, I think it's about 10%, 8 to 10% of those infected go on to develop the disease. So that's just my education. My career history is not as straightforward. And so I've kind of outlined what I did, and what the stuff in the brackets is what I took from the previous job. So after my undergraduate, my Bachelor's of Science, I decided that I needed to work for a year or more to pay off my student loans. So I worked as a laboratory technician at Connaught Pharmaceuticals, which was a biotech company in Toronto, where they made vaccines. In fact, they were making acellular vaccines for whooping cough at that time. And so that laboratory technician job came about because I had undergraduate research experience, so I got a job right away. So that's where the undergraduate research experience really served me well. as a laboratory technician, I was, I had to do whatever my supervisor told me. So he told me to do the experiments and so on. So I didn't have as much input in the direction of the research. So I thought after a year, I've saved enough money to pay off my student loans. I will go to graduate school. So when I was in graduate school, I was a research assistant. I had my own research project, but I also did work on other projects that my advisor wanted me to, because I was supported by actually a National Science Foundation award. I also had the opportunity to teach, and so I learned to teach. And there, as a graduate student, my research experience and my lab work as an undergraduate and as a lab technician helped. So after I got my PhD, I had a little baby, and I thought, well, I really don't want to do a postdoc at this time, but I need to have some way, some money coming in. And so I did contract teaching in Montreal again, and we have a college system, and I taught at those colleges as an instructor, and I taught physics and chemistry. So basically, first year physics and first year chemistry at the university level. And there, my experiences as teaching assistant really helped. Then my family moved to Vancouver, Canada, and the college instructor jobs are very far and few in between. One of my friends suggested that I look into intellectual property. So there was an opening for a patent agent at a law firm close to where I lived, and I went in to speak to them. And they were very interested in the molecular biology, biochemistry, biotechnology knowledge that I had, and also the writing skills that are needed to write your dissertation, write your papers as a PhD student. So I worked as a patent agent for about a year, and I realized at that time that I did not really like the dry writing that is needed. So legal writing is even drier than scientific writing. So I thought, okay, well, I really like the science, but this writing is a little dry, and I would really like to generate my own intellectual property instead of filing patents on other people's intellectual property. So then I thought, okay, I will go back and going back into science, and I became a postdoc. So I had a postdoctoral research position at the University of British Columbia. And there, of course, my research experience and even my patent agent experience helped. I was funded through a biotech company as a postdoc, and my postdoc research did generate some patents. At the University of British Columbia, I was in the medical school, so I had the opportunity to volunteer to teach. And so I said, why not? I have had some experience, so I taught as a problem-based learning instructor in the medical school. And so then after 3 years, it was time to start looking for a permanent position. And so I applied to a bunch of places, and I had a bunch of interviews. And I chose to go to California State University, Fresno, which is in the same system as the one Charles Brooke went to, CSU Chico. It is also an ag school. And we also have an enology program where we make wine. So I took that position partly again because it was a two-career thing. So my husband got a job with the the city government, and I had a position at the university. As a professor, I taught, in fact, a lot. I taught courses in microbiology, intro bio, molecular biology, and medical micro. I also had a research program that had undergraduate students and master's level students that was funded by the National Science Foundation, as well as the National Institutes of Health. My research was on the stress responses of bacteria. So instead of just working with mycobacteria, I started working with Staphylococcus aureus, aureus, Bacillus subtilis, and also I went back to my PhD organism, the algae, and in this case, blue-green algae, cyanobacteria. As a professor, the third thing that you do, in addition to teaching and research, you do a lot of service. So you serve on a lot of committees. You are involved in a lot of initiatives. So I served on committees at the department level, college level, university level, and even the CSU system level. And I also did service to the wider scientific community. I reviewed papers. I reviewed grant proposals from many different agencies. Some of those were international. And I served on panels, the National Science Foundation panels. In fact, when we have these review panels, we usually ask whether anybody's interested in serving as a rotating program director. So I was asked that question, and as a rotating, they wanted to know if I was interested in as a rotating program director, because I had a broad expertise in microbiology, biochemistry. With biochemistry, you can do plant biochemistry, animal biochemistry, microbial biochemistry. And so I said, yes, why not? And so I was a rotator for 2 years, and I was asked whether I would like to apply for a permanent position. I said, okay. And I applied for a permanent position, and I got it. So I've been a program director for about 8 years. And as a program director, I also served as a cluster leader, which is similar to a department head. And I had a chance to serve as a science advisor to the associate director for biological sciences, excuse me. So let me tell you a little bit about what the National Science Foundation does. So the mission of this agency is to fund basic and fundamental science that really promotes the progress of science, advances the national health, prosperity, and welfare, and secures the national defense. We're a pretty small agency. We only have 4% of the research and development funds of all of the research and development funds from the US government. This was in FY 2020. I work in the Biological Sciences Directorate, and here you can see that we have 4 priorities in fiscal year 2024, and one of them is building a resilient planet. So what does that mean? That is climate change. How does the warming climate affect all forms of life? What are the feedback loops that instigate further warming? And can we harness biological mechanisms and process to mitigate climate change? The other major priority is advancing the bioeconomy, and you may know that President Biden had released an executive order on the bioeconomy last year, and this is why this is a main priority for federal funding agencies. And advancing the bioeconomy, what it does is it advances science to use biology to develop novel products, either wholly biological ones or those that are based on biology. And of course, some of these products would help with climate change. So the circular bioeconomy feeds into climate change research. We are also interested in integrating across biological sciences. It is really hard to be an expert in all biological sciences. So people have become really siloed. That's why we are prioritizing integration of biological subdivisions and trying to encourage cross-disciplinary work to address grand challenges in biology, such as organismal resilience to global climate change. And the last, just like other directorates at NSF, is committed to the development of a science and engineering workforce that draws on the talents of all Americans. So to build this workforce, we support researchers at all career stages. We fund, teachers who teach at high schools and elementary schools. We fund undergrads through research experiences in undergraduate sites. We fund graduate research fellowships. We fund postdocs. We fund assistant professors. We fund associate professors and so on. And also, we fund HBCUs and tribal colleges and EPSCoR states. So what do I do as a program director? Well, I was hired as a microbiologist and as a biochemist. And so my job is to support innovative, basic research, basic and fundamental research in microbes. And I want to support innovative and merit review activities in basic research that contribute to the mission of NSF, which is, improving the nation's technical strength, security, and welfare. So a program director solicits, receives, and reviews research proposals. They make funding recommendations. They administer awards. We influence the direction of science. We draft and shepherd through the clearance process things like white papers, solicitations, dear colleague letters, and other different documents pertaining to innovative research. We also perform a lot of outreach to academic research communities. I go to meetings. I was at the ASM microbe meeting. I went to the beneficial microbes meeting just 2 weeks ago. And we have meetings with our colleagues in other agencies and across directorates. We also mentor. We talk to people who are submitting proposals. We try to help them find a home for their proposal and give them feedback. So we're mentoring potential and current investigators. So how does this relate to climate change? So I'm a little further away from climate change in terms of actually doing the research right now. My job is to fund climate change research. So I do that as part of my work in core programs. So I'm assigned to core programs in the Integrative Organismal Systems division of bio, and the programs that I've been associated with are symbiosis, infection, and immunity, plant-biotic interactions, physiological mechanisms and biomechanics, integrative ecological physiology, plant, fungal, and microbial development. So all of these, my work is finding microbial research in these core programs, get the best review possible, and find the best research to fund. Then, because there are microbes in almost everything. So microbial research is throughout National Science Foundation. You find it in the Geological Sciences Directorate, you find it in the Engineering Directorate, not just bio. So we have special programs. Some of those are across programs, across divisions, across directorates that are interdisciplinary. One of them is Global Centers. This is also an international program, youth-inspired research addressing global challenges in climate change and clean energy. That was in fiscal year 2023. And this was in collaboration with the British, the Canadians, and the Australians. Organismal response to climate change is Integrative Organismal Systems program, and we are going to release a specific microbial component to that in a few weeks. Something like building synthetic microbial communities for biology mitigating climate change, sustainability, and biotechnology. That is also across the Bio Directorate. And then when I was science advisor in the the Biological Sciences Directorate, one of my jobs was to serve as the bio representative in the Climate Change Coordinating Committee. So other than just, doing the administrative work that is involved in program management, I'm also given 50 days to perform independent research. So my lab is now closed. I'm an emeritus professor at CSU Fresno. They needed that space. So I really can't do any research there. So I do research with my collaborators. And this allows me to maintain involvement with basic research. So I work on cyanobacteria still. I'm looking at the role of thiols in the environmental stress response, especially the response to ocean acidification, to oxidative stress. I have started a collaboration with methanogens in the gut microbiome. So this is in the human gut microbiome, and this is with Christine Moisel-Eichinger in Graz, Austria. And in fact, I'm going on sabbatical in a couple of weeks to spend time in our lab, actually being back in the bench doing actual research. So I'm very interested in the role of microorganisms in global climate change. This relates to my experience as a faculty where I studied stress responses in bacteria. So what does my day look like? Charles was good enough and he didn't have a slide. Well, so, my day is like, there's emails, lots and lots of emails from potential PIs, from colleagues. I have lots and lots of meetings with colleagues, potential principal investigators, collaborators. I'm involved in review of proposals, so I have to set up panels. I have to find reviewers. Once we give awards, I am involved in award management, so I need to be really aware of how much money we have, and so having some budget expertise helps. There's a lot of reading, keeping up with the literature. There's a lot of writing, solicitations, white papers, DCLs. There's a lot of editing. And so all of this keeps me pretty busy, and each day is different because I also go to meetings. I also, am doing some of my own research, and depending on what the deadlines are, for example, if there's a panel that has to happen within a couple of months, then I have to do a lot more work on that and less work on other things. So I can't tell you what an exact day looks like. It changes very much. The other question that we were asked to answer was why, what are the reasons for this career? So I think one of the common themes in my career is this real excitement about basic and fundamental research. I think we need basic and fundamental research to have translational research, to have applied research. So we want to reduce methane emissions. We have to know what is going on at the microbiome level with the archaea and the bacteria and the interactions. So those are the sort of things that I've always been interested in. Always been interested in very basic science. So that's one thing. The other thing is NSF allows me not only like a depth in a particular science, but breadth. I get to talk to colleagues in engineering. I get to interact with my colleagues who are geologists. I get to interact with education people. I get to, I'm involved in a lot of different things across agency, across like federal agencies. I was involved in the microbiome interagency working group, and so it gives you a real good overview of the state of science in the United States and internationally. And I like that, I like seeing that. What is needed? What can I help advance? What can I push forward? And in microbiology, and also the role of microbes in climate change.
Thank you, Dr. Rawat, for the nice presentation.